So before we start, this is cake 1, and it's the overview of protein synthesis.
This is an entire cell. This is the nucleus where the DNA originates and where the mRNA is created before it moves to the ribosomes to create.
And this is cake 2, and this is a zoomed in structure of the nuclear pores. And here is where the RNA strand comes out. And this is the DNA strand, and this is the DNA strand with the complementary RNA nucleotides.
One important thing to note about RNA is that instead of thymine, which is the base on DNA, it has uracil, which will be noted by yellow candies.
So these are also the RNA polymerase represented by the black jellies. So first, our first step is transcription. To begin, basically we have an RNA polymerase, which is the enzyme that binds the DNA template. And this will catalyze the formation, and it will bind to the promoter, which is the section on DNA that once it binds, it will start transcription. So the RNA polymerase will start transcription, and the DNA will start to unwind, and the hydrogen bonds between each base will break, but we don't need to break them. And once they break, each separate DNA strand will go separate, and
individual RNA nucleotides will
come on one by one, eventually creating a full strand of mRNA.
And these are attached by hydrogen bonds, and the RNA nucleotides are attached by covalent bonds.
Basically,
They attach in the same way that DNA nucleotides would, with the hydrogen bonds and covalent bonds, except for the difference of the bases and the sugars that are used.
Just to form a nucleotide is a structure with one base, one phosphate, and one sugar. And this could be represented by the base, the phosphate, and the sugar is in there. But in the textbook picture, they didn't show it, so we thought it wasn't necessary, but it's there. So moving along, once the RNA strand is made by all the RNA nucleotides, it reaches the termination signal, and this means that it's going to tell the nucleotides, oh, stop bonding, you guys are done. Therefore, the RNA strand will now split apart from this DNA.
And the two DNA strands will come back together.
From before, the one that split before.
Reforming this.
And the RNA strand will go into transition.
And it'll go into the nuclear pores, so it comes out of the nuclear pores. And so we have our codon here, which is AUG, and it
it's also known as the start codon.
Yeah, so tRNA comes in with the anticodon and the amino acid. So the anticodon would be UAC, and the amino acid would be methionine. And then they bond through hydrogen bonds. And this is a ribosomal subunit. This is ribosome.
The ribosome travels along the mRNA and helps to attach.
Yes. And then the next tRNA comes with the corresponding amino acids as well as the anticodon. So they come, and then they bond together as well. And then the ribosomes shift. So they shift here, and the amino acid here detaches the tRNA
and goes to find another tRNA with the same base pairs to do the process again.
And the tRNA leaves.
And then the amino acid is binded to the amino acid on this one to form. Oh, yeah, okay. So then
to form bipeptide bonds, so they're bonded together.
And to show the process of how the ribosome, the RNA will kind of move with the tRNA is so once this happens, you have that. And then the third...
No, this one leaves.
Yeah, this one will leave. And then so the next anticodon will come right here, and these will bind, and the tRNA will then break off of this, and the amino acids will transfer with they will
bond. They'll detach with the They'll detach off the tRNA, and then they'll bond with the amino acid.
This tRNA is gone, and the ribosome will keep on moving.
No, these are, and then these leave.
These two leave.
Yeah.
These stay.
And now you have Yeah. And so, that one.
And then it reaches a stop signal, and at the stop signal, it tells the ribosomes to stop adding more anticodon and amino acids. And then after that, the amino acid detaches off the tRNA
To form.
so it forms this string of amino acid, which is amino acid, and then they fold on top of each other to create proteins.
Which will be used for whatever functions, yeah. And
then
These will all, the mRNA, the nucleotides will detach and they will be recycled again to make another mRNA.
and the whole process goes on
It's
to make proteins.
a circle.
Thank you.